All right. Last chapter, chapter 20, which is on viruses, bacteria, and archaea. Basically, what we're looking at here is the differences between the two viruses and bacteria. We're not going to get much into archaea. Viruses, there are a lot of different types, but you can see here bacteriophages. These are viruses that attack bacteria. And then this is like a virus that attacks humans. You can see this is the influenza virus. They're not really considered living. However, Hank Green made a TikTok recently that he made the claim that he thinks they are living, and there is debate on whether they're living or not. But they're very small, and if you take a look here, the virus is very small compared to a prokaryotic cell, which is very small compared to a eukaryotic cell. And remember, you're made of trillions of these eukaryotic cells, so these viruses are very, very small. Classification, there are two parts. First part is one of 7 groups that the scientists kind of separate based on their genome, and then the second part is just a very similar to our classification. Even though they're not living, they still classify them into this kind of format. So again, they're too small to be seen under a microscope. 3 main parts. They have the size and shape, so like how big and what the shape is, the type of nucleic acid, whether it's single or double-stranded, or whether it's DNA or RNA, and the presence of or the absence of an outer envelope. That's just the outermost portion here. The capsid, you can see here that's that protein coat. This protects the DNA inside. You can see that the different types of bacteriophages and whether it has a capsid, or the capsid. The envelope is it's like the outside that's made of a membrane and has this glycoprotein, so it makes it look like a host cell, or makes it look like a cell just in general. And that's if it has it or not. Glycoproteins we already talked about, but just a reminder, these are like the keys that allow the virus or whatever these are what essentially allows cells to intake things. But these are like keys, and what the virus does is it tries to use these glycoproteins to get into cells, and this is almost like a Trojan horse kind of deal. Now, we've talked about this a little bit when we talked about the COVID vaccine and how viruses spread. But basically, the virus tricks the receptor cell into allowing it in. The virus then releases its genetic material. This causes the cell to make that virus, or make proteins that will make that virus. And once the cell gets filled up with these viruses, it will burst. And then the viruses that were made inside the cell will go out and try to infect other cells. You can see here, this is a bacteriophage. It's also known as just a regular phage. If you ever hear phage, that's just a bacteriophage. And this type only affects bacteria cells. So this is what was in Jimmy Neutron, Carl's stomach. So it really wouldn't have attacked Carl and them because it only attacks bacteriophages. Now, there's 2 different types of I would say reproduction within viruses. The lytic cycle is when the virus injects bacteria, which quickly replicates in the virus and destroys the cell. The lysogenic, is when the DNA is injected. However, it doesn't kill the cell, but the DNA is going to fuse within the bacteria's DNA and becomes dormant. Bacteria divides and then creates more bacteria with the viral DNA. So essentially, this is a less deadly, but it just incorporates its DNA into the host genome. So it's like becoming part of the host, so it will always produce that type of virus. Retroviruses. So this is incorporated in the AP exam. So this is really important that you understand. This is the same thing what we just talked about with the lytic versus the lysogenic cycle back here. I can't get here. All right, here we go. Let's the lytic versus the lysogenic cycle here. But basically, what's going to go on here is the same thing happens just within your DNA, and the DNA the virus that has RNA, and then that RNA gets transcribed, translate, however you want to say it, back into DNA. So what happens is your DNA then gets fused with the virus DNA, and then you'll keep on making that virus. So same thing as a lysogenic process, but in this case, it's for humans. Viroids. These are not viruses technically. But what happens is they're double-stranded RNA, and the double strand comes from their circular nature, and they work in a little bit different way. Basically, they attack a lot of plant diseases. This is what caused I don't have it here, but this is what caused the plant famine back in Ireland like a couple hundred years ago, and they just attack cells in a different way. Prions. This is a very interesting one. This also, I've heard a lot of scientists say that they're scared of prion disease because they are very deadly, and they're it's hard, there's not like an antibiotic or antiviral to really treat them. They're out there. Prion disease, there's no genetic material. They're just misfolded proteins, and these misfolded proteins cause other proteins and to also misfold, which causes diseases in a lot of mammals. Mad cow disease is an example of this, where the brains of the cows just deteriorate. It's very, very scary. All right, everything before this was viruses, and everything after this is going to be prokaryotic cells. I'm going to go through this pretty quickly because a lot of this stuff we've maybe talked about before, but these are just parts of a prokaryotic cell. So the capsule, this is just like the gel coating on the outside of a cell. This helps the cell to adhere to things. It's made of a polysaccharide, which means it's a carbohydrate. The fimbriae, these are just hair-like appendages that jet out from the bacterial cell that will help attach it to different things. Nuclei, this is just the region where the chromosome's found. You can see it's circular here. It's just the region where that chromosome is going to be found. And the flagellum, this is a rotating filament that will help propel the bacteria cell into a new location. It's used for movement. We talked a little bit about plasmids before, so again, I'm not going to spend too much time here, but basically what happens is these plasmids are extra portions of DNA, and what they can do is they can transmit these between different cells. So this is how DNA can get passed from one cell to the next, is through something called conjugation. We can do this. We didn't do the lab this year for this. It's called transformation, but we can do this as well. All right, we have a couple of different morphologies or shapes of bacterial cells. The Coccy is the spherical or circular shape. You can see the Streptococcus here. That's the type of bacteria, and this is what causes strep throat, and this is a circle. Bacillus, this is the straight rod, so this looks like a pill. Lactobacillus is the example that I give here. You can see how it looks like a pill. Lastly, Piruli or Spirilium or Spirilia. These are just like a corkscrew or like a curved and spiral virus. These aren't as common, but the Borrelia burgdorferilia, I forget how you pronounce the last one, but it's what causes Lyme disease. You can see it right there. All right, with bacteria cells, they grow really rapidly because of exponential growth. Basically, the cell just keeps on doubling and doubling and doubling. For the first few rounds, it's very it's not as there's not much of an increase. But after, when you get like millions and millions of cells, that's where you're going to see this really dramatic increase. But this is capped off by limiting factors such as nutrition or space, where there's just not enough stuff for the bacteria cells to grow. All right, we're going to talk about how bacteria cells grow and what environments they need. We have certain type of bacteria cells called obligate anaerobes, which are unable to grow in the presence of oxygen. And that might not make sense to you because everything you know needs oxygen, but this type of bacteria needs a place without oxygen. You can see what tetanus does. You can see this locking of the back. But basically, this type of bacteria needs an environment where there's no oxygen. Obligate aerobes, these are bacteria that require oxygen to grow. Basically, just what the type of bacteria you would think of. Facultative anaerobes are able to grow in either oxygen or absence of oxygen. Greater growth in oxygen. These are just they don't care if there's oxygen around or not. They don't need it, but they'll grow in both. And lastly, aerotolerant anaerobes, bacteria that can grow in either aerobic or anaerobic environments. Very close to the last, but just a little bit different. Tooth decay is one of these. South Park had an episode on tooth decay. You can see obligate aerobes, they need oxygen. Obligate anaerobes, they don't need oxygen. And aerotolerant, doesn't matter. But facultative anaerobes, they like oxygen, but they can grow in either. All right, pathogens. So bacteria secrete these toxic materials sometimes. Sometimes they're even included in their cell walls. But basically, they try to kill the host in some way, and that's how bacteria can be deadly. To give you an example, like babies aren't supposed to be given honey under the age of 1 because there's a bacteria cell that grows within the honey sometimes, and that bacteria cell will secrete a toxin that the babies can't digest and kind of break down until they're about 1, when their immune system and their gut flora mature. So this toxin is really deadly to babies under 1, but not to older people. Antibiotics, this is a substance that either inhibits or kills the bacteria cells, and they do it in a couple of different ways. You can see where they inhibit the cell wall, or they stop with some biochemical or biosynthetic or metabolic pathway, and you can see how they kill the bacteria. These antibiotics were not always known. This is large portion of people died in wars up until we knew about antibiotics. Think about the Civil War. When you got shot, you got infected. I mean, that was a very big killer of people back then. Now we have these antibiotics, but now we're also having the bacteria evolve around these antibiotics because evolution is always occurring, and we're finding more and more bacteria that are resistant against our antibiotics, which is a big problem because after a certain time, if all our bacteria are resistant against all our antibiotics, we won't have a way to treat them effectively. Gram staining, this is a type of procedure that allows scientists to identify different types of bacteria, because it's really hard to understand without doing these tests how to identify a bacteria cells or bacteria culture, because there's a lot of different types of bacteria out here. Basically what they do is they use staining to determine whether there's a gram positive or gram negative. Gram positive, these are thicker cell walls, single membrane of peptidoglycan turns purple. Gram negative, thinner walls, there's a double membrane, and the peptidoglycans between the membranes, this turns pink. We usually do a lab with gram staining. Again, I don't think we'll get to do it this year, but it's a fun lab and something you'll get to later on. All right, this is the end of the chapter.